what I'd like to do now is introduce you to Dietrich Stephan. He is the founder and CEO of Silicon Valley Biosystems. I promised you that this morning you were going to get a little bit of different perspectives on this topic, and indeed, Dietrich is going to help us really kind of look at some of these issues from the private sector, and where is the potential, and where this industry is heading. In particular, what I'm excited about is that he is going to be able to speak a little more about bringing the genome to the point of care, which is extremely important when you get right down to it. So welcome, Dietrich Stephan.
Thank you, Susan. Well, good morning, everyone. What a wonderful conference. I have really been impressed with not only the quality of the speakers, but the format. Really letting people expand into an hour has been refreshing in this age of tweets and 15-minute talks. And I'd also like to thank Gianrico Ferrugia and Scott Beck for putting in place what I believe is the premier applied individualized medicine program globally. And in my wanderings, I've seen a lot of people use that moniker as a brand enhancement for their healthcare system, or try and approach it in a piecemeal way. But they really have built something that's real and something that is impacting patients today. So it's applied. And for that, I'd like to thank them. And all of us have been working tirelessly for the last few decades, actually, with this dream that the genome would be applied at the point of care someday, and that we could practice medicine with both eyes open to better improve patient outcomes. And to see it come true and is amazing. So it's our golden age, if you will, and we should take a breath and appreciate it. So thank you to the SIM team. What I'd like to talk about today is a little bit of how we've played a small role in bringing some of the tools and strategies of individualized medicine to the point of care so that patients can benefit, and hopefully so we can reduce healthcare costs at the macroeconomic level, although that is a more difficult, interestingly enough, more difficult challenge, not only to project but to measure. Again, I'll start by telling you a little bit of context as to why I have been focused, laser focused in one specific area for the last few years at sort of a broad-based level, and then go very deep on the technical side so that there's an appreciation for how hard it is. And you've gotten a lot of that during this conference, so this won't be new, but I think what may be new is how do we take the incredible innovation that you've heard from the speakers at this conference, who are largely working within ivory towers at high-powered institutions with hundreds of millions of dollars to throw at a program. How do we take that innovation and drive it into the community-based setting where 80% plus of us get our care, so that everyone can benefit from these new tools and strategies? And so that type of scalable infrastructure is what I'd like to give you a sense from, and it's a different type of activity than tinkering with one patient where you have immense resources, where you have a 30-person tumor board, where you have a slew of high-seqs at your disposal. It's a different endeavor. So that's what I'd like to focus on a little bit. So let's see if I can figure out how to move this. So this has been our mission collectively for the last 20 years, and as explicitly stated as our mission statement within our diagnostics company. It is to enable the clinical community to bring the genome, the one-shot assay, cheap and accurate, to the point of care where patients are actually managed instantaneously, and to improve healthcare globally, meaning it's inexpensive and scalable. So there's a lot embedded within that one sentence that we need to grapple with. And it's all predicated on a world that we still believe in. And it's coming true before our eyes, but there's a lot of work to be done. We still believe that we're going to get a cheap genome and that it's going to be accurate. And hopefully Illumina gets some pressure, so give Matt Pissard some difficulty on their pricing here. But we need a sub-$100 genome in order to get this technology to become pervasive. I still believe we can get there in a few years, not a few decades. When we get there, I still believe that individuals will be sequenced either at birth or in the first trimester, because there's immediate value that can be unlocked from that now cheap assay, and we can continue to unlock that value across an individual's lifetime. Where will that genome reside? The natural place is within the electronic medical record. It is where all other sensitive medical data resides. Why shouldn't the genome reside there? It's true already that doctors walk around with tablets when they're seeing patients, even in the community-based healthcare system that ties into their enterprise EMRs, where all of that clinical data is stored. I believe that when a physician is sitting in front of a patient and entering in the ICD-9 or 10 codes, or the clinical signs and symptoms, that there will be a role for a software system to do genome-based diagnostics instantaneously in that environment and return that genomic information back to that tablet right there, consistent with the clinical workflow at low cost. Again, diagnostics in the software layer based on an archived genome that we carry around with us. Obviously, this breaks down a little bit with respect to some of the somatic applications that you've heard about, but for germline disease, I believe in that world. And if you do as well, then that's what we're building, and I'll tell you a little bit about that. Let me start by telling you a little bit about the applications that we envision. And we don't envision that genetics is a niche tool. We believe it is a tool that is relevant to every single human disease. And the way I categorize human diseases is maybe a little different than we usually think about them. Usually we think about them around specialties. I tend to think about them in terms of the type of diagnostic or the type of clinical workflow into which you're trying to shoehorn that diagnostic. And so I have 5 categories that I usually talk about. They include monogenic disease and hereditary cancers as one bucket. These are things that, these are mutations that you inherit from your parents that are highly penetrant, meaning if you have the mutation, you generally get the disease. And we have understood how to test for them and deliver that information for the last 30 years. These are diseases like cystic fibrosis, Duchenne muscular dystrophy, and about 4,000 other Mendelian diseases, including another 100 or so tumor suppressor genes that, again, you're born with and that will dramatically increase your risk for cancer in your lifetime. So why is this an application area or a unique field of use, if you will, for diagnostic genome testing? It's because the mutations have unique characteristics. For loss-of-function mutations, you have a blown-out loss-of-function mutation on both alleles generally and a recessive mode of inheritance. For dominant mutations, you can characterize those types of mutations, and we have a huge reference set of mutations that have been collected, again, over the last 30 years against which to train. We have a robust testing infrastructure in this country, and it keeps getting better with companies like Veronata and Counsel and others. And the types of tests here are carrier testing, PGD IVF. We now have circulating fetal cell testing that can pick up mutations, mostly aneuploidies today, but hopefully we'll get into resequencing for Mendelian diseases in those circulating fetal cells in the maternal bloodstream soon. And we have actions. We have counselors that understand how to communicate this information, and we have reproductive options of various flavors for people who are at risk. This area is going to continue to grow because now we can do in one test every Mendelian disease as opposed to trying to understand from family history which single gene to test for. And so this is an explosive area. You're seeing many companies, Good Start Genetics, Council Genetics, Verinata, Ariosa, Sequenome, become billion-dollar companies in this space, and it's quite impressive. Second clinical area is in chronic diseases, and this is an area that I've personally grappled with, and the reason is very strategically because while monogenic disease we understand, it still only accounts for between 5 and 10% of disease burden globally. Whereas chronic disease is what we're all going to get and die of, quite frankly, and it accounts for 80% of disease burden globally. 80%, that's interesting. We all thought that infectious disease was a big issue. We've done really well with infectious diseases over the past 50 or so years, and that has spawned this huge extension of lifetime and this massive epidemic of chronic disease, which now is what is bankrupting our healthcare system, quite frankly. And so very strategically, in 2005, and I'll tell you a little bit about that, we tried to understand how we could practice precision prevention, which would stratify the population as to their hardwired genetic risk, again, genetic, this is genetic, so they could modify their environmental triggers for these diseases. This is going to be, probably, in my opinion, probably the single most impactful area in healthcare in the next 50 years, but it's going to take that long because we need to flesh out gene-environment interactions, we need to generate the actuarial data that'll support reimbursement, et cetera. The third clinical area is germline pharmacogenomics, and I think we've all understood how valuable that is over the last few years. Actually, we've known about it for a while, right? We had the Roche P450 chip, remember that? And no one bought it. And so everyone realized it was important. No one bought it. And I think it was because the use case, the drug-gene correlations were poorly understood. It was disruptive to the clinical workflow when you're sitting there writing a prescription to say, oh, well, maybe I'll get a metabolism check on the P450 chip. Let's wait 2 months. By the way, who's going to pay for it? And so that really didn't work. And now finally we're starting to see an uptick. And you heard some of the wonderful work that is happening here at Mayo Clinic with respect to the EMRs triggering a notification that's in big red letters saying you got to get this genetic test because there's some important information here. The fourth area is really in cancer pharmacogenomics, and we've seen the wonderful work that Foundation Medicine has done and the success that they've had recently around testing for molecular markers in tumors and trying to couple those to therapies. Our big challenge in this area as a community is to bring that type of testing up to the point of diagnostic biopsy when the tumors are not chemo-resistant, perhaps less multiclonal, and try and break or understand how we can layer combination therapies on top of the standards of care, which is no small challenge, especially if you're talking about unique combinations for an individual which have no precedent or evidence base around them. So I think that's the big challenge in oncology, but it's also the big opportunity. And you have folks like the CEO of Millennium saying that's where we need to go, but how do we get there? And Foundation Medicine is a great first step, but hopefully over the next decade we'll see more progress to that front. And then finally, host pathogen and host graft interactions. We've known about transplant and HLA for a while. We're starting to understand the interactions between pathogens and the human genome. We know some people are resistant to HIV infection because of a polymorphism. We are starting to understand how the microbiome interacts with our germline genome and other things. And so this is really how I break down the space. The final area is trauma, and it's not on here because some people say, well, that's not a genetic disease. Some other people would argue, well, perhaps there's a reason that that person had that traumatic event, and there's certainly differential wound healing characteristics, et cetera, between individuals that might have a genetic flavor to them. But in general, I leave that off of my slide. So I've gone through sort of an evolution personally as to how can I be most impactful in this space and how can I add value. And really, I started off life really intrigued by the applied nature of genomics, and particularly of the Human Genome Project, where in the research setting you could find a broken gene and you could turn it on in a diagnostic setting the next day, as long as you showed that that variant was not present in 200 normal chromosomes. You could get an Athena or someone else to turn it on and test patients for it. That was very intriguing to me, and I said, well, great, research, that's where it's at. And so spent a number of years contributing on the research front in really two formats. One was, and it's worth looking at this in a retrospective way so we see sort of as a field where we've come. In the late 90s, in DC at the Children's Hospital, we said, all right, there are all these clinical problems that kids have downstairs. What we should do is take the top of this hospital and put in place a translational research program with scientists who know about all these fancy tools, and we'll go downstairs. Scientists will go downstairs, ask the doctors what they really need, go back upstairs, turn on all this machinery, figure it out, and then give it back to the doctors downstairs. And so it was this beautiful, nice little circle, and everyone said, yeah, that's what we'll do. And we built out the 5th floor and hired 100 people, and it was the reality of it was nothing that we worked on upstairs made its way downstairs. Great example was, we went downstairs, we figured out that a portion of kids with medulloblastoma never disseminated. They didn't need destructive chemo and radiation, which left the whole population with their pituitary glands knocked out, growth stunted, deaf, and mentally retarded. So how could you figure out the half of people that just needed their tumors removed and spare them that damaging therapy? So we did a wonderful expression signature. We validated it, Nature Genetics, 90% classification accuracy. And then we said, well, okay, great. Well, it's published. Maybe someone will read it. But certainly that signature is not in play today. And we just thought it was going to osmose its way back downstairs. This is a real issue and was very frustrating back then. And so this was before IVD-MIA, before Randy Scott really blazed a trail with FDA, before we understood how to validate across very large independent cohorts, and really how to communicate this type of information to physicians. I mean, I think one of the key learnings from genomic health was that none of these tests are binary. They're not black and white. There's a spectrum. And if you present a spectrum to a physician, or even a patient, we'll get there in a second, they freeze up, right? It's like, well, where do I land on the spectrum? If you now break that into 3 buckets, green, yellow, red, and say, okay, well, if your patient's in the yellow, we're not quite sure what to do. But if it's in the green or the red, now we have a decision to move forward with. That's what sort of freed up that expression test for Oncotype DX, at least in my naive understanding, and allowed that to become a pervasive test. So those types of things were not available to us back in the late 90s. So then had another opportunity to try and be more applied in Arizona, where we set up an entire biocluster focused on translating genomic discoveries into the hands of physicians and patients. And it was wonderful, big effort. But at the end of the day, there were pieces of the puzzle that we didn't put in place that simply blocked the ability to translate those results in an effective way. And so I would say 95% of the innovation that happened there during my reign never made its way into the hands of patients and physicians. And it was really because of the things that are listed below research on this slide. So validating a research discovery rigorously, getting regulators to say, yes, it's okay to turn that on in a clinical setting. And that often includes educating regulators, because they're just like everyone else. If they haven't read the paper and they're not up on the latest bleeding edge fancy tool, they have no idea what it is. And so there's an educational process there. What are the ethics around turning some of these things on and appropriate use? We grappled with this a lot in the early days around common risk variants for chronic disease. And we had very clear boundaries, but other people had different boundaries. And you know, how do you standardize? Physician education and adoption is absolutely non-trivial, and in fact, that is what most biotech companies spend the vast majority of their dollars on. It's a sales force, it's educational materials, and it takes a lot of money, tens of millions of dollars, to get through that phase in the translational pipeline. Setting policy, you heard Ed Abrams talk a lot about how hard it is to create the data sets and have those conversations. And then finally, reimbursement. These are absolutely non-trivial, and I would argue they're an order of magnitude more complex than simply discovering the correlation to begin with. So I left research and I said, well, you know, there are thousands of people with NIH behind them doing this research. What's really missing in the world is a vehicle to deliver the genome to the point of care for those 5 clinical verticals. So in 2005, a colleague of mine and I started this company. It was a company called Navigenics. And we said, well, this is going to be absolutely disruptive to clinical practice as we know it, because by and large, no one was using genetics in clinical practice. And so we put an incredible team around this. We put David Brailer, the first healthcare IT czar for the United States, because we knew, like I said, we knew this was going to eventually go into the EMR. We put John Doerr, the guy who cracked the business model for Google. We put Sue Siegel, the current head of GE Healthy Imagination, and a whole set of individuals around this, because we knew we were going to be taking fire because we were trying to transform healthcare. Healthcare is a big monstrosity, and this is a tiny little company trying to make a difference. And where we started was, this was at the time where we still had a $100,000 genome. So we couldn't start with the genome. So we took a representation of the genome and we said, all right, what are the clinical areas that representation is relevant to? Where can we actually provide accurate and actionable information to patients and physicians? And it was in the area of chronic disease risk and in pharmacogenomics, and that's where we started. And the plan was we were going to ride the cost curve down and add those 3 other clinical areas: monogenics, cancer pharmacogenomics, and host pathogen interactions as we sort of segued into a genome over time. Great plan, great team. We put $100 million into it. We were too early. And what we decided to do was sell this company to a company that had the fortitude and the balance sheet to keep it going long enough so as to be able to slip into that, let's say, $100 genome and layer on top of all of those clinical verticals. And so this went to Life Technologies, now Thermo Fisher, and Ronnie Andrews, I believe, is using this as a platform for rolling out their diagnostic portfolio. And so fingers crossed, this will reemerge as something interesting. In parallel, because this is now out of my control, and because we've finally gotten to the point, and you've all seen this slide, where we're at $1,000 exome and a couple $1,000 genome within current reimbursement paradigm price points, if you will, we continued to innovate in a different form and format, and this is Silicon Valley Biosystems. So we thought we were going to get $1,000. We thought we were going to get a couple $100 exome by 2014, and again, because Illumina has no pricing pressure, that is stalled out. So hopefully, in the next 12 to 18 months, other technologies are going to come online, and we're going to continue to drive that price to the floor on the sequencing side, so we can get to a pervasive technology. But $1,000 exome is great, and in fact, we can get hundreds of dollars at the gene panel level. And so you heard a lot about the wonderful HNPCC panel work that's ongoing here at Mayo. And so a lot of people who are trying to do DNA-based diagnostics are segueing to panels because it's cheaper. And so we're also starting with panels and exomes. And this is the current state of the world with respect to adopting next-generation sequencing for molecular diagnostic purposes. I would say it's safe to say that every DNA-based diagnostics company has had the light bulb go off where they've said, well, wow, we can test more genes for the same price. And by the way, we can also reduce our internal cost. And in the face of reimbursement pressures, CMS not paying its bills for the first 2 quarters of this year, people have all sort of decided to supplant Sanger with NGS-based technologies. And when I say all, I mean people like LabCorp, Quest, ARUP, Mayo Medical Labs, and the whole second tier of DNA-based MDX companies. They've all done the back of the envelope math, said we're switching to NGS. Many of them have bought equipment, and many of those people are now grappling with the assay. And so this is the classic Jeff Moore technology adoption curve where you have early innovators jump in first, and those people bought machines early. Then they have to grapple with these assays, either an exome or a genome or a targeted panel. And this is absolutely non-trivial to get working at clinical grade, and I'll talk about that in a minute. Then you have to extract the patient's variants from that aligned genome as compared to the reference genome and figure out what all those variants are. Are they polymorphisms or mutations? So you have to annotate them with the world's body of information, meaning DB SNP, 1,000 genomes, et cetera. And then you have to put them through a classifier to decide which one of those is relevant to the clinical trait of interest that the patient sitting in front of the doctor asking about. And then finally, you have to figure out how to report on this so people actually believe the results and can act on them. So where are we? We're about right here today in this technology adoption curve. So as I said, many to most people have bought NGS technologies. Some of them, a small amount of them, have gotten the assays working at clinical grade, and I'll define what that means. But the vast majority of them have not gotten past that, and this technology is not yet in broad-based play across community-based healthcare, right? That's the message here, is we still have a long way to go on just the assay before we can start laying on top of that these rule sets that I talked about. So what's involved in this new technology backbone? A lot. And in fact, we as a company said, all right, well, we're just going to take a genome, right? It's digital, it's accurate, and move forward with these 5 clinical areas and provide these application areas for clinicians to invoke on their iPads at the point of care. We had to go all the way back and basically figure out how to get next-generation sequencing to work at clinical grade. And when I say we, this is us with a bunch of different partners, and it's absolutely non-trivial. And then we had to say, well, okay, well, we're not gonna go do all of this stuff and then turn on a clinical application area that's not relevant to the marketplace. And so we did a strategic analysis of where revenues are flowing in today's diagnostic market. And as you can see here, this is a preprint from an article that's coming out, Personalized Medicine, in November. We decided the first application area to start in is Mendelian disease, single gene disease, and the second is in tumor drug selection, the third is in germline pharmacogenomics, the fourth is chronic disease. Remember, that's where we started 5 years ago, and now it's fourth. And then the fifth is in host pathogen graft interactions, because there's little science around that today. And so that's what we did. So we said, all right, we need the assay first, we need it to work, and then we're going to segue into all of these 5 verticals over time. And that's how we decided to build this company. So why did we have to grapple with the assay? It's absolutely non-trivial. This is what we used to see in a diagnostic environment with Sanger-based sequencing. A master's level CLS could look at this trace right out of the machine and say, that's a mutation as compared to the control. I'm going to put it into my clinical report, and I'm going to hand it to my laboratory director, and it's going to go out the door. This is a representation of what things look like today out of these machines. And that same person just locks up. They can't move forward. And so we said, and oh, by the way, the performance characteristics of that are very different. So we said, all right, let's, again, bring a great team to the table who can help solve this. And so the first team we put next to us was a team that's very good at building information technology companies. This is Sequoia Capital. They've built Oracle and Cisco and LinkedIn and a number of these sort of big IT companies. And the second is the Center for Individualized Medicine, the Mayo Clinic, recognizing that they are the #1 clinical brand in medicine, but there's substance under it, and they're actually leading the way in individualized medicine today. And so we said, all right, well, let's bring some really smart people next to us to help us figure out the assay. And oh, by the way, perhaps with the help of the third largest diagnostics company on the globe, we can together really get this to be pervasive, which was, again, our mission is global in nature. So this is what we've built over the last 2.5 years. We've built the ability to sequence at high quality, turn that into a representation of a patient's genome, which is the alignment and variant calling step, extract the differences from that patient with respect to a reference patient, so now that's the variants, understand which variants functionally impact the disease, the genes within which they reside, as opposed to polymorphisms. So now, what are the functional variants within a patient's panel, exome, or genome? Turns out there are a lot. It's why we all look different, and in fact, we all carry actually dramatic mutations, mostly in a recessive state, 100 or so. So then the question is, well, how do we link that to the phenotype that the patient has, or the question they're asking across those 5 application areas, and then instantly deliver that information in an easy-to-understand report. It's non-trivial. I'm not going to go through all of this, but there are many, many modules. These are computer software modules that take the output of a sequencer and turn them into a final report. And each of those has many knobs on it that you can tune and dial. And the list goes on and on and on. And in fact, it continues to grow. People come out with new aligners and variant colors all the time, for example. And so the first thing we decided to do was to build an entire new operating system for next generation diagnostics. We call this Cloud Keeper. I won't go into too much technical detail. What I'm trying to describe here is that as you scale outside of an academic medical center, you need to ruggedize this stuff and have an environment for rapid development, clinical validation, and then implementation. It can't be the bioinformatics person that's sitting on their desk drinking coffee who works from noon to 9 p.m., right? That's not what this is. So this was an entire new operating system. It's called Cloud Keeper, and it's now running in the Amazon cloud, of course, in a HIPAA-compliant way where no PHI is actually pushed into the cloud. We're still grappling with whether the genome is PHI, and maybe we could have a good debate about that as a community over time. Formally, it is not recognized as PHI by CLIA and CAP. So, alignment and variant calling. This is non-trivial. We thought, well, the machines, they output it, you put it into the Broad BWA GATK package, and out comes the variants. In fact, there are a multitude of new variants, variant callers that are coming out. And this is a wonderful paper by Ra et al. With a quote that says, "The perfect and most appropriate parameterization is not often available or easily obtainable," meaning it's really hard to find the right aligner and variant caller for clinical work. That's how I read this. And in fact, there are a ton of them, and they continue to grow. So which one do we use? Which one do we pick? So we had to build an entire benchmarking environment. And this was actually done in collaboration with the Mayo Clinic, with Jean-Pierre Kosher and Eric Klee and others, where we basically built a benchmarking environment where every one of these we can put in and benchmark versus every other aligner and variant caller and understand its sweet spot. And then once we have its sweet spot, we can plug that into an amalgamated or patchwork solution that performs optimally across the entire spectrum, across the entire mutational spectrum. Just to give you a sense, if you take BWA GATK off the shelf as it's tuned when you download it, it is not appropriate for clinical work, period. You have sensitivities in the 70% range. Short read sequencers, 100 base pair, 200 base pair reads. Sanger's at 700. And so if you say, well, we're going to use NGS because it's cheaper and we can test more genes, but we miss mutations that exceed the length of NGS read length, you actually have decreased sensitivity relative to the gold standard. And I would argue that that is an immoral trade-off, if you will. And so how do you see mutations that exceed the read length of this NGS-based technology and get you to the current gold standard. We put a lot of development into this. We're not quite there yet, but we think there is a solution to do this at clinical grade. And I won't go into what that is today. But how does this benchmarking engine work? And again, this is data generated together with the Mayo Clinic, although they did these tests independently from us. You know, these are not our numbers. These were generated on Mayo samples with Mayo scientists. If we take 3 types of tests, the first is genetic data into which has been seeded synthetic mutations across an entire spectrum of types, single nucleotide variants, insertions and deletions, and variants in highly problematic regions of the genome, meaning repeats, homopolymer tracks, dinucleotide, trinucleotide repeats, etc. How well does our amalgamated patchwork solution work in finding variants? Remember, we're back at the beginning of the assay now. This is crazy. We have this big vision, and we're grappling with the very foundation of these assays. So we've finally gotten this working at what we believe is clinical grade, meaning above 99% sensitivity for standard single nucleotide variants and indels within the read length of the NGS machines. Highly problematic regions we're still having trouble with. But then, by the way, so does Sanger. And so as, you know, we think we're comfortable with this spec, but we're going to continue to iterate here until we try, you know, hopefully get it back up to 99%. But then again, as long as we're calling this out sort of on our label as if you have a mutation in a highly problematic region, our sensitivity of detection is 96%, that's going to be the path forward. The second test is, these are hereditary colon cancer patients with known disease-causing mutations, sequenced on NGS technology, and our algorithms were applied to it. How well did we do? Standard SNVs and indels, 100% sensitivity. And then in a subset of those patients where they were fully sequenced across all genes in that panel, I believe it was 15 genes. So we know every mutation by Sanger. How well did the variant caller, aligners and variant callers work relative to that expanded set of now gold standard variants? It did well, so above 99%. So we feel pretty good about the very first step in this assay. How do we take it forward? And again, I'm not going to go into it as excruciating detail, but hopefully you're starting to get a sense for the rigor that needs to be applied to out-of-the-box NGS technologies in order to get it into the clinical space. So annotation, we had to aggregate the world's body of variants, publicly available variants. This is non-trivial. I mean, scientists and researchers have been contributing to public databases for 30 years and have had no incentive to do it correctly. And so you see dbSNP polluted with mutations, you see HGMD polluted with polymorphisms, you see all these fragmented locus-specific databases, some of them tucked away in companies that you'll never access like Myriad. And so how do you bring all this together so you can apply it to a genome and do this type of diagnostics at the point of care? And so we've downloaded the ENCODE data set, and we've had to build new compute systems. Remember, we're trying to do this at scale, so we are annotating everything in memory, and for the computer jocks in the room, what that means is that the entire database is in RAM. And in fact, SAP has a new computer, came out after we developed this, called HANA, which does the same thing, and they're trying to sell it into the genomics marketplace. But basically, we can do 600 million rows of annotation on an exome in about 15 seconds. Now, if you think about trying to do this at a population-wide level, you start to get some comfort that you can do it as opposed to hours or days across a large sample set. The variant databases are noisy, so our annotations are noisy. So how do we train the machine to look at a new variant and decide whether it's a polymorphism or a mutation? That's non-trivial. And so we had a training set of mutations and polymorphisms, about 140,000 of them, 70,000 polymorphisms, 70,000 mutations. We trained a classifier on that, and we said, all right, now we're going to give you another 140,000. You're blinded. Tell us which is a mutation, which is a polymorphism, again, in this Mendelian disease application area. And it actually did pretty well. And so we have really nice performance around what we're calling our pathogenicity prediction engine. So why is this relevant? I'm sorry, I'm going to bring it back up again. Why is this relevant? Patients that walk through with Mendelian disease and try and get testing, show no paucity of new mutations, meaning there are new mutations at an extremely high frequency in the molecular diagnostic environment, and interpreters don't know what to do with that. They call it a variant of unknown significance, and they apply very crude prediction technologies to it, SIFT, polyphen, SNPF, et cetera, to get some inkling as to whether the report that goes out says, you're going to get breast cancer, or you're okay. And what inevitably happens is the report goes out saying, we have no idea what this new variant does, and good luck. That's exactly what happens. It's a routine finding, and the slope of that remains constant. So it's a big problem in diagnostic environments. So we said we want to solve that, because if we want to be truly relevant, we need to provide higher diagnostic yield. And so this is a simple illustration on the left-hand side of a training set, about 80 patients, about 800 or so variants, where we knew what was a mutation and what was a polymorphism, and we applied our classifier versus the standards out there in the world, and we do pretty well. This is a colon cancer example, so hereditary tumor suppressor gene blowout mutations. This is CFTR, where we really had a set of samples where we knew what the pathogenic variant was and what the pathogenic variant wasn't. And just to cut to the chase, if we see a variant of unknown significance, with sensitivity and specificity in the 90s, we can say whether that causes the disease or not, as opposed to when you get down in the SIFT or polyphen range, where it's essentially random information and really has no use. Okay, so now it looks like we've got the technology working pretty well. We've got the assay working at clinical grade sensitivity, clinical grade specificity, again in the 90s. We've got the ability to say, if we find a variant in a gene, that variant with, again, confidence intervals around it, is likely causative of the disease or not. How do we deliver that out there in the world and put that final piece of the puzzle in place? So what we've built is an alpha version of Diagnostic Studio. This hasn't been rolled out yet commercially, but it allows true subject matter experts to go in and see the results of a panel, an exome, or a genome, and say, that's the one. You know, I know that that's the mutation based on 30 years of clinical experience, and because I've seen every patient in the world with this disease, that that's the variant. The computer, missed it. It's the number 2 on the list. I'm going to click on that. What happens then is the report is automatically issued, and that expert learning feeds back into the system. So the next time we see that mutation in a patient in some other geography, we can provide increased confidence that's a true clinical variant. There are other systems that do this. I think Gene Insight is the one out of Sandy Aronson's shop that has gotten some traction. But we feel that this is a nice way to capture expert learnings and really create a constant learning environment. This is what the report looks like. It's 3 pages. It's clean. I'll give you a couple examples. CLIA has no idea how to regulate this new diagnostic environment, not only on the NGS side, but on the in silico diagnostic side. So we had our CLIA inspector come in, and the first thing they asked is, where's your lab? Because there's no sequencers in our dry lab. It's computers. We have, our technical validation. Usually you run it multiple operators, multiple days. Now the computer simply runs it, 100 times or 1,000 times, and you see if there are any glitches in that process. It's different. CAP understands this much better, and we hope to be CAP certified at the end of this year or in early 2014. And they have some nice guidance around it, but they're still evolving. So how does it work? We're just commercial stage. We just got our regulatory stamp. But in the background, we've been testing this, and you've seen some of that data. let me just give you a couple of clinical vignettes, and I know you've seen a lot of these, and these aren't new, but perhaps it'll tell you that, you know, we're in business. So this is a great story. This is a couple that couldn't get pregnant. They had IVF 3 times. They had a son the first time, a son the second time, and then twins in their 3rd pregnancy from the same IVF. The first son started showing signs of language regression, seizures, IQ drops at about 2 years of age, was originally diagnosed with autism, went through a whole battery, Stanford, you name it, and then started showing signs of neurodegeneration, got all the testing for that. In fact, it was sort of 3 years, $300,000 later, and was still in decline with no diagnosis. The second child started showing clinical signs that were identical to the first child. And finally, they reflexed to us for exome sequencing. We immediately dropped out a compound heterozygote in the TPP1 gene, which is a rare form of Batten disease, infantile Batten disease. Delivered that diagnosis, but then matched that patient to 2 clinical trials, one ongoing at Columbia, which is a viral gene therapy replacement. You know, it's delivered into the brain. The other is a BioMarin drug that was in early stage trial and true enzyme replacement in California and in the UK. And so we got these kids into the UK trial, and my understanding is they're stable now. So that is a wonderful story. Interestingly, we got biochemical validation after we found the gene through exome sequencing. Orthogonal validation, but usually you do biochemistry first. The second case study, and this happens to be from the same clinician who sent us an exome again, was a complex 8-year odyssey. I won't go into all of these things, but it was a movement disorder. In fact, they had suspected Rett syndrome initially and went with a FISH test, which came back clean. And then 8 years of testing, all kinds of doctors, they finally came to us. We did an exome, and they have a single nucleotide mutation in the MECP2 gene. So they actually do have Rett syndrome, but the wrong test was ordered. And so this starts to make you think about, where does this type of test fit into how we perform molecular diagnostics today, and how is all of this going to reshuffle itself once we have a cheap genome at our disposal? And I would argue that in certain use cases, we should be ordering an exome out of the box today. So for example, in extremely genetically heterogeneous diseases, for example, in pediatric neurology, epilepsy, deafness, etc., why would you serially walk through genes, often dozens of genes, at X hundreds to thousands of dollars per test, instead of going right to an exome? This is a great example. Both of these are great examples of that. And what are the economics around that? And how do we flesh that out at the policy level? We're at a new stage in this challenge, I think. So let me just stop right there, but just to tell you, this was great. This doctor, Michael Ches, said, your testing seems to find things that nobody else's does, and this is, after going to GeneDx and Baylor and everything else. And I think it's because of the focus that we've put on quality, sensitivity and specificity, and then making sense of the variants that come out the back, not just sending a list of 100,000 variants from an exome. So hopefully this is a piece of the puzzle that can really fuel the efforts that all of you are doing in your ivory towers, if I may be so bold, and drive it into the community-based setting so that everyone could get access to this wonderful new technology. So our mission is to enable. We feel like we're solving a pain point in the marketplace. We're clinical, not research. It's a different thing. It's a different thing. Community. We want everyone outside of this group to be able to access this technology. The genome. We want it to be cheap. We want it to be accurate. And we want it to be pervasive. At the point of care, we need to engage EMR companies. We need to understand how Oracle and their system can house this information, how it talks in a bidirectional way with EMR systems, and how these new decision support tools work in that context. And then how do we provide the applications on top of this platform. I'm using that terminology very specifically. I'm trying to make the analogy between a ubiquitous technology platform that's accessible to all of us and diagnostic applications that sit on top of it in these 5 clinical areas. And then finally, how do we drive this globally? And again, we're thrilled at the potential opportunity with Mayo Medical Labs and some of our other partners to really enable this. So thank you, and these are the people that actually did the work.
Dietrich, thank you so much. It's really remarkable to hear about some of the investments that have already been made in this effort. I wanted to just ask, sort of return to a theme that we've hit upon in a number of conversations over the last few days, because I think listening to you and others, there's this sense of there's just so much, and how can the practicing physician possibly keep up with all of this?
I think you hit the nail on the head as to one of the biggest problems that we face as a community that has invested so much time and money in developing this massive set of new technologies and strategies, right? We have this wonderful tool set available now that this group as thought leaders knows about, understands, meaning what's real and what's not. And the challenge now is to drive it to the masses. Mm-hmm. That is an absolutely non-trivial problem, and we're working on it from just one small, through one small lens, which is the genome, and how to bring genetics forward to the point of care in a very simple, easy to understand way across all clinical use cases. Now, there's a whole other world outside of genetics, meaning, what do I do then? What's the right drug? What are monitoring tests, for example, or what are the right screening tools if we know someone's at risk? And I don't know what the answer there is. I think there is a need for a new body that evaluates and blesses these new strategies by specialty area and is real time, says, okay, here's the latest thing, it's real, here's how you get paid for it, here's when you should use it. Mm-hmm. And that should happen sort of on a monthly basis. That doesn't exist yet, and so physicians are completely overwhelmed, and in fact, you could argue that they're in violation of their Hippocratic oath because they should be taking the time to understand this stuff, but they can't.
Sure.
So it's a big problem, and I don't know what the answer is.
Mm-hmm. Mm-hmm. Well, hopefully some of our conversations over the last few days in the hallways are getting at some ideas for that as well. Let's take a look at a few of the questions that have already come in. There's one here specifically on does your sub $100 genome include the cost of analysis of that data, the storage, etc.? This questioner observes, rightly, that some of the other presentations that we've heard from suggested that was on the low side, that there are additional costs. And is your company working on that piece of it as well?
We are. When I refer to the sub $100 genome, that is just for the assay. That is my hope for just the assay component, recognizing there are all of these other pieces that need to be stacked on top of it and paid for in order to sustain, the genetic testing enterprise. And certainly storage is one component of that. We are, as I mentioned, moving everything have moved everything to the cloud and are using storage resources like Glacier to fulfill our storage requirements under CLIA and CAP of X years that we have to save that data.
And that's a cost savings.
That's a cost savings. Mm-hmm. But interpretation is another big piece of it. Right. And so our company hopes to get paid in the X tens to hundreds of dollars for every interpretation we do on top of that assay and on top of the storage. And so you start to see how these things stack.
Sure.
But yes, we're dealing with it. We're as best we can, and we're going to ride the cost curve on the compute side down as well.
And I think this is a trend that we're seeing more broadly in, for instance, med tech, and in some instances other pharma companies, where you no longer simply sell a product, you have to also sell kind of the services around it, the analysis, the storage, the kind of the extra brain power that you bring to the information.
I think that's spot on, and the most obvious area there is there will always be a need for a subject matter expert looking, at this data and blessing it. For example, a molecular pathologist or anatomical pathologist that is deeply steeped in that disease which is being reported. And how do you capture that in a scalable way is a big question.
I notice we're getting close to our break time, so let's see if we can rapid fire through a few more questions. Will it be cheaper in the future to repeat the sequencing rather than paying for long-term storage of the data? What do you think?
Maybe in 5 or 10 years.
Okay, 5 or 10 years. Good, quick answer. You highlighted 6 different challenges. Of that list, challenges to integrating genomics, I hope that's not anything personal. They want that coffee break, I guess. Of the 6 challenges you highlighted to integrating genomics into the clinical practice, what do you consider the most critical to success? Can you prioritize those?
Every single one of them is critical to success and needs to be grappled with in a way that is analogous to hand-to-hand combat. I mean, this is really this is an on-the-ground battle on regulatory, on reimbursement, on policy, and it's why it's taken us 20 years as a community to start to see some traction.
Final question. I think this is so great because it's wonderful at these gatherings.
Is that me?
Is this one of our, not sure, what, is that the bell for coffee? Tornado warning, somebody thinks? Okay. Final question, because I'm really excited to have an entrepreneur here on stage. I think it's such a great, fresh perspective for a largely scientific gathering. If you had to start a new company in this space today, what would you do differently?
Well, we fully intend this company to be the delivery vehicle for the genome at the point of care. So I will not start this company again.
Okay.
If I was going to start another company, it would be precisely to fill the burning need that you articulated early, which is how do you evaluate this tsunami of new technologies and strategies, and how do you package them appropriately to maximally impact an individual's care at the time they're presenting for that care. And that's a really big challenge, the integration of those things. And that doesn't exist yet.
Mm-hmm. Well, I hope that maybe you and Ed and some of the other folks gathered here might put your heads together. Maybe that's going to be your next endeavor, Dietrich. We now are, before we thank this speaker, I'm going to let you know that we do indeed have that coffee break, and it will go until 10:30. If you can resume then, we'll have our 2 final speakers, and please thank Dietrich Stephan.